cancer is and make a tiny introduction about this topic for them talking about oncogenomics.
The term cancer englobes a large group of diseases that are characterized by the development of mutant cells which divide, grow, and spread in an uncontrollable manner through all parts of the body. In a normal cell, it divides and dies um, during a programmed period of time called the cell cycle. However, the cancer or a tumor cell loses that ability to die and divides almost without limit. And that multiplication in the number of cells form masses called tumors, which destroy and replace normal tissues, and it doesn't do his main function. Next.
Okay, I'm gonna talk about a history. First, I wanna give you like a fun fact. I don't know if it is a fun fact, but yes. Um, First, the first time that, like, the first fossil tumor that people found was in Egypt, and it was like many years ago. Was I think there was a woman with a tumor in the cranium, and they also found a script that said what was happening in that moment. So like they didn't talk about cancer, but they talk about like what was happening and what was the patient feeling. So that's something like for me was really impressive, because like cancer is since like many years ago. um Okay, then the first time that the word cancer was used was in 3,000 years before Christ, and it was in Egypt. So, yes. Then Hippocrates, I don't know how to spell that name, Hippocrates, in 460 and 370 before Christ. He was known as the father of the remedy, and he spoke the first time about carcinoid and carcinoma to describe the formation of tumors. So, like he took that like that word because carcinoid is related to crab, so like the crab growing, so like they had a really similar growth, so he wanted to relate it with that. Then in 1665 became like it was Anthony van Leeuwenhoek with the microscope, so like they could investigate one cell by cell, like to describe the difference with morphologies about one normal cell and one tumor cell. So they start like knowing what was different about like with both cells. Then Rudolph, which show I don't know how to say that. In 108,000, he founded the cell pathology, and like he started investigating like most like with the microscope and what was happening. Like he said like, okay, this is something that is wrong, that is like killing us. So he wanted to investigate more of that with the help of the microscope. And yes, Then, like many years since that, we have like a much more technologies and much more things. Like we can amplify the genome to identify tumors, cancer, and what is happening. But and then in this moment, like we are all developing more things to know about this. So yes, that it's like a point of the history. Okay, now I'm gonna talk about different tumors. We have two different types of tumors. One is benign and one is malignant. Like the benign, like the main difference between these two tumors is that the benign like grows slowly and don't spread around all the whole world, the whole body. Sorry. And the malignant like grows really, really, really fast and can spread to other tissues, other organs. So this is called metastasis. So like that is the main difference about benign and malignant tumors. I don't know if I passed.
Yeah,
Do you?
yeah, there
Okay.
it is. So there are many types of cancer, but mainly they differ by solid tumors and liquid or blood cancer. Solid tumors also divide in many other types of cancer. For example, there is carcinoma, melanoma, sarcoma, and these have different etiologies and different origins. For example, a carcinoma comes from epithelial cells, melanoma comes from melanocytes on the skin, and sarcoma comes from mesenchymal cells. And then there are the liquid cancers that divide in lymphoma and leukemia. Lymphoma comes from lymphocytes, and leukemia comes from immature blood cells. These subclassifications are already being developed. I don't know how to say that, but with the genomic advances, we have new molecular classifications that divides different types of cancer by their molecular origin, for example, or the pathways that they come from, and stuff like that, that are fundamental to developing new treatments and making like cancer less horrible that it is actually.
No.
Yeah.
Okay, let's talk about causes. Cancer can have many causes because almost everything you see can cause you cancer. First, the chemical cause. When you are in direct contact with dangerous chemical substances like tobacco smoke, nitrosamines, polycyclic aromatic hydrocarbons, alcohol, asbestos fibers, benzene, and many other substances, you have a high risk of developing many different types of cancer, like lung cancer, and larynx cancer, brain cancer, many types of cancer. And second, if you don't do exercise and if you have obesity and a bad diet, you can interfere with your metabolism and make it damage your cells more than it does normally. And if you don't do exercise, you cannot have a strong immune system to fight that back. if you have an infection, a viral infection, you know that the viruses can enter fragments of DNA into your own genome, and it can shut down many of your own genes. And if you shut down one of your proto-oncogenes, you can develop cancer later. The ionizing radiation is not a particularly strong mutagen, but if you have a continuous exposure to radiation, you can develop leukemia or skin cell. The most famous one is the sun, the damage caused by the sun. And physical agents, if you have a repeated burn on the same part of your body, of your skin, through the process of healing, the excessive cell proliferation can cause cancer in your cells, just repeated injuries. Also, autoimmune diseases can cause cancer by damaging the DNA in surrounding cells when you have an inflammation because of the cytokines. Okay, next.
Other thing that is important to know is that even the use of some cancer drugs have an impact on causing cancer. For example, I had a friend in school that had a lymphoma, and she was treated by chemical therapy, by chemotherapy, and this chemotherapy cured her lymphoma, but caused a leukemia, and finally my friend was affected more by the leukemia and than the lymphoma. So this is what developing new treatments talk about and are needed for.
So you just need to live to get cancer. Do you see?
Okay. Also there are a type of cancer that is hereditary, and it consists of when you have a specific syndrome. That syndrome can make you more prone to develop cancer. If you have, where we have here many examples like HBOC syndrome or Lynch syndrome or Li-Fraumeni syndrome, and all of them share the same characteristic. They are caused by mutations in proto-oncogenes that are inherited from the parents. So when you are born, you have, in fact, more probability of and damaging your other allele in your life. Okay, next.
So this table is here to show you like the context of cancer, how many people are affected by it by year, and which of the types of cancer have like affect more people. So the mean diagnosed new cases for each year is like about 17 million, and like at every moment there are like 90 million people affected by these types of disease. So it's huge in the world. Okay, so as many people know, the traditional treatments for cancer is surgery, bone marrow transplantation, chemotherapy, and radiotherapy. But we are developing new types of treatments, such as hormone chemotherapy, targeted therapy, and immunotherapy. But what happens here, as I mentioned before, for example, chemotherapy has huge cytotoxicity to our normal cells, and this makes secondary effects huge, as in many patients and many cancer patients. So all of these alternatives offer a more specific treatment that or that targets cancer specifically and increase the effectiveness of the treatments and are becoming more and more effective than the traditional treatments.
Okay. I'm gonna ask you guys a question. If you remember something about the about yesterday that some partners talked about genomics.
Yes, Maria Camila. What is the specific question?
Like, I wanna know if like the class remember what was genomics, like in contest, like something general.
Okay. What is the meaning of genomics?
Yes.
I don't know if someone want to speak about it.
No one. Really?
Tengo que responder en inglés.
Yes. It's an English class.
Okay, I'm going to try. Genomics, in general terms, is a subfield of the genetic. That's what I get about yesterday.
Okay, that it's okay. So yes, that it's a good like meaning, but and I'm gonna add something is that genomics is the characterization and quantification about genes. So oncogenomics is the same thing, like they wanna characterize and quantify, sorry, the genes related to cancer. So, like they wanna know like the mutations of the genes, what is happening. They do that with the transcriptomes and epigenomics. So they wanna know or they wanna improve the diagnosis of cancer and mutations, like to improve the therapies, to improve like if someone like sorry, I'm gonna start again. They want to improve the diagnosis, so they want to know earlier if someone have or not cancer, so like to treat that quicker. So also they want to have like better treatments, like David said some minutes ago. They want to have less secondary effects, so they want to investigate. They're investigating, they're searching, they're doing a lot of things to know like cancer treatments, diagnosis, like like to people don't die like that fast, or they can like be good if they have cancer. So yes.
So basically, cancer presents as a complex system that involves many variables in its origin. So these types of tools, such as oncogenomics, serve as a huge help for knowing what Camille was saying. And to give you more of a context about the origin of cancer, as everything in life, cancer has a molecular and genetic origin. This involves the cell cycle, and you can see this cycle as a process with many checkpoints in which there are many genes involved that check whether the process is going well or not. These genes are divided in oncogenes and tumor suppressor genes. So in general, cancer may come from the activation of the upregulation of oncogenes or from the downregulation of tumor suppressor genes. Next.
No, I can pass.
So the oncogenes are briefly that have the potential to, they come from proto-oncogenes that are genes involved in the regulation of the cell division, but activated, mutated, or upregulated, and get these abilities to create cancer. So, for example, there is this RAS on the right. There is the RAS gene that creates RAS protein that is involved in this cell division and has like a cascade of signals that make survival or apoptosis of the cell. So up here, this gene is considered an oncogene, but when it's damaged, it becomes a a proto-oncogene. But when it is damaged, it becomes an oncogene that can cause cancer and is related to many types of cancer. If you look for RAS protein, it has a lot of studies behind it. So here it's kind of important to make like this type of facts. Oncogenes express as dominant diseases because you only need one damaged copy to express cancer because it doesn't downregulate the cell division but upregulate it. So one of these copies it makes up for the disease. Then it is Theodore Boveri, a very important scientist that in 1902 and 1913 started proposing about the origin of cancer that was in the chromosomes and that had like external factors involved in it. So when he proposed this, many of his colleagues didn't accept it, but years later, in 1969, when the term oncogene was developed, he was proven right about what he was talking about.
Okay, now I'm gonna talk about tumor suppressor genes. Okay, these are the genes that reduce the chains of cells turning into cells cancer. So these are genes that are found in the cells and they control the production, of excess cells. So if there is a mutation or a deletion in these genes, like the cell division cannot stop, like the I'm sorry, the cells cannot respond normally to the checkpoints that have like the cell cycle. Okay, so if this happens, like the cells are gonna divide and divide and divide, and no one's gonna have stop them. So these are the ones that are in charge of like stop and make the apoptosis when they found like something wrong in the DNA. So if there is something bad in that gene, like the mutation is gonna continue, and they're gonna grow and grow, and the tumor is gonna be like formate.
So this is an example of this origin of cancer. In this case, it's in retinal cancer, in a retinoblastoma, that you can see that when the origin is from a tumor suppressor gene, the cancer expresses as a recessive disease, because you need both copies to be mutated. Like in the first example, the child with both normal alleles needs both copies to be heated or to be lost to get cancer. Because if you only have one bad copy, the other copy is making the function of the tumor suppressor gene work well. And this is the second example. When you inherit one mutated copy, you need to get the other one a mutation for getting cancer. That's
Okay, here we have three examples of drugs that are used to treat cancer. They basically are made to attack or to block or to inhibit proteins, part of the cell cycle or of the or growth factors. The first is called well, imatinib in a scientific manner. That what you see in the screen is the artistic name. Imatinib works by stopping the tyrosine kinase, uh, and by stopping that enzyme, it can slow down the growth or finally result in a programmed cell death, right? In certain types of cancer cells. It is used to treat leukemia, gastrointestinal tumors, and breast cancer sometimes. And it is a simple molecule, relatively. The second is called scientifically as trastuzumab. It is used to treat breast cancer and stomach cancer, and it works by binding to the HER2 receptor and slowing down cell duplication, right? It inhibits the protein of the cell cycle. And the third is called formally as bevacizumab. It is used to treat a number of types of cancers and sometimes eye diseases. It is a monoclonal antibody that works as an angiogenesis inhibitor. What does that mean? The angiogenesis is the process of growth of new blood vessels, of creating new vascular endothelial inside of the cancerous tissue. So the bevacizumab slows down that growth of new blood vessels by blocking the growth factor VEGFA. Okay, next.
So, as we all know, there are many oncogenomics efforts that many people and a lot of resources in them. We are going to show you two examples of this. The first one is the Cancer Genome Landscapes. That is a project that was looking for or made as a product the landscape of mutation between many types of cancer. I think like 15 or something like that. And it involves a lot of people and has a huge product that we are showing you here. What kind of mutations are involved in these types of cancer, and which has the most mutation, where they are located, and many things like that. And next. The other one is the Cancer Genome Atlas. I think it is the biggest ever in cancer, and it analyzed over 11,000 tumors from all over the world that included 33 types of cancer. Sorry. And this analysis provided deep understanding of what happens in tumors in humans, how they appear, where they appear, and why. And this, like the main product you can see from this project is that this team published a lot of very important papers about the classification of cancer and many things like that, and they are involved in, or they are related in something that is connected with systems biology, because they are, or they did a network of these different pathways that cancer follows by joining different omics, like as you can see in the picture on the right, like transcriptomics, epigenomics, genomics. Everything was joined together to get huge networks of pathways that help us understand better the way these diseases appear, and that's it. It's huge. You can look up this project, and it's really impressive.
Okay, investigated, we found one article that personally was like really impressive. Like, it is not just like about genomic, but it's like microbiome with genomic to identify a cancer. So like, it was really impressive because they, like I said before, like scientists want to know earlier when it's like a cancer in a patient, like to treat it faster. So in here, they could know if there was a cancer, like really fast, just by taking samples of blood or other tissues and analyzing if there was like some microbiome of bacteria or viruses. So like, this is really impressive because like they took samples of people that they know that they had cancer, and also they took samples of people that were like okay, and then like they quantified them like with the genome, and they found that they had some like things of microbiome in their genome. So with that, they then could know if they had cancer of no, like the patients. I don't know if I let me understand, but it was really impressive.
And finally, well, we can see like the actual benefits of using these tools, these oncogenomics. And there are three examples that the theme of the TCGA show. The first one is type of breast cancer has been divided in different molecular subtypes that right now are being managed differently in the clinic. So you get a different treatment for what was the same type of cancer before. Right now is more specific for what you need and what can cure you. So that's pretty interesting. Another thing is the therapeutic targeting of some mutations. In this case, a metastatic melanoma, that is one of the most aggressive types of cancer, has improved the treatment because of because of now we know the mutations that occur and we can target them, be specific for cancer. And finally, well, the lung cancer that we saw earlier was the one that affected most people on the world. Is being treated with also target agents that are specific for these types of disease. And that's it.
Thank you for your attention. If you have any question, let us know.
I have a question.
Say it.
Go ahead.
Did you know or did you read something about cancer treatments using synthetic biology, like with modifying virus or other microbes? Because as you know, we're going to talk about synthetic biology, and we read that they're using modifying viruses to kill cancer cells. But I don't know if they are already using this technique, or if it's just like a hypothesis or a method that it's like starting to grow. So I don't know if you read something about this.
I read something about, not specifically that, but about biological therapies that involve not chemicals or not physical kinds of therapies, but biological things. The main force at this moment is immunotherapy, that is involving like blood cells, for example, white blood cells that they the cancer. They are like I don't know how to say this right, but it's like pumping people blood cells that can fight these types of cancer, and that is pretty interesting. But I didn't know about synthetic biology. I don't know if Cami or John said something about it.
I really
Well,
don't
yes.
even read about that.
There are, I read that there are some efforts to use CRISPR-Cas9 as a useful tool to treat cancer, because you can just repair the gene that is damaged, that is causing you a tumor. If you use CRISPR in a very delicate way, you can maybe repair the tumor cells and cure some types of cancer.
Okay, thank you.
Thank you very much for your presentation, guys. Any more questions?
Yeah.
I have someone.
First of all,
Mariam.
Congratulations to my brave friends to for make that presentation in English. I'm very proud of you. I understand everything you say. Well, the question is if you know or you read about some strategy nowadays about the gene P61 or some gene that is related to the control the dead cell, the program dead cell, because there's treatments about viruses and all that stuff, but what about the genes or proteins that control the cell device, bueno, the cell division? Or if the teacher knows.
Yes, they are called the oncogenes. Make up oncoproteins, and these are like the proteins that have the ability to cause cancer. But in this case, specifically, you were talking with P53, I think.
Yes, that is related with the tumor suppressor genes.
But it's
Exactly.
when
So
the treatments about suppressor tumors is when the gene are already damaged. Talking about to prevent that damage in the protein or gene. Do you know?
I don't
okay. No,
know.
I
Say it.
go ahead.
No, you.
Okay. Right now, I only know that when it is already damaged, but John talked a little about this genetic therapy earlier, so I don't know if he knows something about it, like for prevention rather treatment.
Prevention. Well, there are many ways to prevent cancer, like using sunblock. I don't understand the question.
You talk about syndromes. That is some cancer. Yeah, you're I know hereditario. I don't know how to say that. And so
Parents.
you already have the gene. You know, you are probably, I don't know how many times to get cancer. So how do you prevent and from from the cellular way, not your habits, not your diet, not your exposition.
But the thing is that like that happens when the gene suppressor it's like had a mutation or a deletion or something. So I don't know how to prevent that because it's something like about a gene that is if it is mutated, like then like you cannot repair that. So if like the causes can be like related to that. So if if you prevent that causes, you can be like you can have a less like a probability to get cancer. But as we
Yeah.
said before, you just need to live to get cancer because like it's a mutation. So it can happen in any time.
So it sounds like you have
Uh,
the treatment if you already have the mutation.
yeah,
That
exactly.
sounds.
So I think that's a really good question, but right now I only know about treatment with genetic therapy, but I don't know about preventing with genetic therapy. I don't know if the teacher has something to say about that.
Yes, I think the best way to combat, I don't know what the term, the cancer is the prevent, is the only way now. But our time is over, so voy a cambiar. Entonces, yo creo que tenemos que darle paso al próximo grupo. Primero, pues agradecerles a los chicos de este tema, pues por primero, pues darse a la exposición en inglés. Eso es pues muy remarcable y además pues es un buen ejercicio aquí. Pues no estamos calificando un inglés perfecto ni nada de eso, pues la idea es que tengan oportunidad pues como de practicar. De todas maneras me quedaron ahí varias preguntas que después les haré pues como en el taller Ya, pues, si además alguien tiene otra pregunta, pues nos la puede dejar aquí también en el chat. Entonces, muchas gracias y adelante el siguiente grupo.